Today's lecture is on quantitative inheritance. In a quantitative inheritance, there are two subpoints. First, that is the polygene inheritance concept example. It means kernel color in wheat. And second one, that is the population genetics. Under that one, we'll see the Hardy-Weinberg's law. First of all, we'll see the polygene inheritance concept example. It is the kernel color in wheat. In a polygene inheritance, there are several genes combined to affect a single trait. It means a group of genes that together determine the characteristic of an organism. It is called as polygene inheritance. It gives the explanations to the inheritance of continuous traits which are compatible with Mendel's law. The first experiment on polygene inheritance was demonstrated by a Swedish geneticist, that means Nelson-Ehle, in 1909 in a wheat kernels. The kernel color is controlled by two genes, each with two alleles. One with red kernel color was dominant to white. He crossed the two pure breeding wheat varieties, that means the dark red and white. The dark red genotypes, that is the capital R1, capital R1, capital R2, capital R2, and white genotypes are small r1, small r1, small r2, small r2. In the F1 generation, the medium red were obtained with the genotype is capital R1, small r1, capital R2, small r2. The F1 wheat plant produces the four types of the gametes. That is the capital R1, capital R2, capital R1, small r2, small r1, capital R2, and small r1, small r2. The intensity of the red color is determined by the number of capital R genes in the F2 generation. 4 capital R genes, that means the dark red kernel color is obtained. 3 capital R genes, that is the medium dark red kernel color is obtained. 2 R genes, that is the medium red kernel color is obtained. And 1 capital R gene, that is the light red kernel color is obtained. That means the absence of capital R gene, it results in a white kernel color. That means the R gene in an additive manner produces the red kernel color. The number of each phenotype is plotted against the intensity of red kernel color, which produces a bell-shaped curve. This represents the distribution of phenotype. While you observe the cross, that means the parental generation, that is the dark red crossed with white. In a F1 generation, there is a production of medium red, and such a F1 generation, when we can self it, ultimately there is a formation of ratio, that means the 1:4:6:4:1. That means that data produces a bell-shaped curve which demonstrate the continuous variation in a wheat kernel from dark red to white in a F2. While you observe this cross, the genetic control of color in wheat kernels, for that purpose you can make the cross in between that dark red crossed with white, the F1 which is produced one, it shows capital R1, small r1, capital R2, small r2, that means the medium red. While you observe the F2s, their genotypes and phenotypes. The first one which is produced one, that is the capital R1, capital R1, capital R2, capital R2, it indicates that their phenotypic character which is the dark red, that means all capital R genes are present, while the remaining 4, it shows a capital R1, capital R1, capital R2 and small r2. it produces the medium dark red. Similarly, these are produced in which there are genotypes, that is the capital R1, small r1, capital R2, small r2. It also produces the medium red. Similarly, the 6 progenies, these are also formed, and in which their genotypes are the capital R1, small r1, capital R2, small r2, and capital R1, capital R1, small r2, small r2. Similarly, small r1, capital R2, capital R2, and all these 6, they produces the medium red. While you observe the 4 individuals in which their genotypes, that is the capital R1, small r2, small r2, and small r1, capital R2, and small r2, they produces the light red. And the one individual in which the smaller one, small r2, small r2, that means there is absence of that dominant gene, that is the capital R. Simultaneously, it produces the white color, and the ratio is 15 as to 1. Finally, the loci that was studied by Nelson-Ehle were not linked, and the genes assorted independently. Later, the researchers discovered the third gene that also affects the kernel color of wheat. The 3 independent pairs of the alleles were involved in a wheat kernel color. That Nelson-Ehle found the ratio is of 63 red as to 1 white in a F2 generations. 1 as to 6 as to 15 as to 20 as to 15 as to 6 as to 1 in a F2 generation. From the above result, the Nelson-Ehle showed that the blending inheritance was not taking place in a kernel color of wheat. In a F2 generations, plants have kernels with a wide range of color variations. This is due to the fact that the genes are segregating and recombinations takes place. The another evidence for the absence of blending inheritance is that the parental phenotypes dark red and white appears again in a F2 generation. It means there is a no blending of genes, only the phenotype. The cumulative effect of several pair of gene interactions gives rise to a many shades of kernel color. He hypothesized that the two loci must contribute additively to the kernel color of wheat. The contribution of each red allele to the kernel color of wheat is an additive effect.